Welcome back. We are in the Chapter 3 entitled Nucleic Acids, Proteins, and Enzymes. Chapter 3 opens up with a question that we should be able to answer at the end of Part 2 of this chapter. The question is, how does an understanding of proteins and enzymes help explain how aspirin works? Before we answer that, we got a lot to get through. First, we're going to talk about nucleic acids. Nucleic acids are polymers, just like complex carbohydrates and lipids that we learned about last chapter. Nucleic acids transmit and express genetic information. There are two types of nucleic acids that we will focus on over the course of this semester. DNA, which is also known as deoxyribonucleic acid, and RNA, ribonucleic acid. The monomers that make up nucleic acids are called nucleotides. Nucleotides have three components. They consist of a pentose sugar, a nitrogen-containing base, and a phosphate group. The identity of the nucleotide is determined by the nitrogen-containing base. 5 types of nucleotides are cytosine, thymine, uracil, adenine, and guanine. Sometimes, and especially in later chapters, you'll see these abbreviated to their first letter, C, T, U, A, and G. Nucleotides differ by their nitrogen-containing bases. Nitrogen-containing bases are either pyrimidines or purines. Pyrimidines are nitrogen-containing bases that have single rings. These are cytosine, thymine, and uracil. Purine nucleotides have double rings, and these are adenine and guanine. I will point out the single and double ring differences between the nitrogen bases in the next slide. The sugar that makes up the nucleotides are either deoxyribose, which is found in DNA, or ribose, which is found in RNA. Here's a picture of our nitrogen bases, again, split into pyrimidines and purines. Notice on the top, pyrimidines only have a single ring, while our purines down here, adenine and guanine, both have double rings. These nitrogen-containing bases are what give our nucleotides their specific identity. Nucleotides bond by condensation reactions to form what are called phosphodiester bonds. This linkage is between what is called the 5' carbon of one pentose sugar, the 3' carbon of the next sugar. I'll explain what that means in the next slide. Nucleic acids grow in the 5' to 3' direction. This will be important when we learn about DNA and RNA synthesis in later chapters. Here's a diagram of RNA. You have your phosphate group, which is highlighted yellow, and your nitrogen-containing base, which is also highlighted yellow. The sugar is highlighted blue here. Counting from the bottom sugar on the bottom left, this first carbon is called the 1' carbon. Then you have your 2' carbon, your 3' carbon, your 4th carbon, your 5th carbon. When the two nucleotides link together in those condensation reactions, the 5' end of one sugar will bind with the 3' end of another sugar. This will continue on and on and on. This is what is meant by nucleotides growing in the 5' to 3' direction. Nucleotide pairs are linked by hydrogen bonds. You can see you have a nucleotide on the left in this particular diagram, which is hydrogen bonded to a nucleotide on the right. Individually, hydrogen bonds can be rather weak, but with many of them together, they can be strong. This is important when the strands become separated during DNA synthesis, which requires a small amount of energy to separate them. We'll also go over that later in the semester. DNA is double-stranded, with nucleotides on both sides of the strand. If you think of DNA as a ladder, both sides of the ladders are a strand. The rungs of the ladder are the nucleotide base pairs. Each nucleotide can only bind with another nucleotide. Pyrimidines only bind with purines. So quick review. Our pyrimidines are cytosine and thymine and uracil and purine. And our purines are adenine and guanine. Within DNA, adenine can only bind with thymine, while guanine can only bind with cytosine. Within RNA, the base pairs are adenine and uracil, in cytosine and guanine. Note that within RNA, uracil is substituted for thymine. RNA is usually single-stranded. It may fold into a 3D structure by hydrogen bonding. An RNA structure is determined by the order of the bases. On the left in this diagram, you have a single strand of RNA. Note that RNA has uracil instead of thymine. On the right, you have a strand of RNA that has folded in on itself. You can see where the bases are paired together. So note that the C's are bound with the G's, and the U's are bound with the A's. DNA is usually double-stranded. As we mentioned before, the two nucleotide strands form a ladder that twists into a double helix. The sugar-phosphate groups form the sides of the ladder, while the bases form the rungs. Here we see the structure of DNA as described in the previous slide. The sugar-phosphate groups are on the side, while the nitrogen bases are in the middle, being held together by hydrogen bonds. Again, note that the A's bind with the T's, and the G's bind with the C's. And this diagram also illustrates that DNA is being synthesized in a 5' to 3' direction. Which brings us to our next point. DNA is said to be anti-parallel. So on the left side here, you have a 5' end, and the opposite of it, you have the 3' end. So one strand is going in a 5' to 3' direction, the other is going in the opposite direction, thus being anti-parallel. So why do we go into all that detail about the structure of DNA? The structure of DNA leads directly into its two functions. DNA, as we talked about before, is the genetic code, which contains all the information for our genes. Its other function is to replicate. The information from DNA is used to copy RNA. This is known as transcription. The information from RNA is used directly to make polypeptides, better known as proteins. In biology, this is known as translation. We'll get into that information in a later lecture, but note that the role of DNA is for replication, and then that information is encoded to RNA, which is known as the process of transcription, while RNA encodes information for proteins during the process of translation. The sequence of DNA base pairing determines the process of DNA replication and transcription. On the top strand of DNA, you have the bases T, C, A, G, C, A, which bind with their complementary nucleotides A, G, T, C, G, T on the bottom strand. That DNA strand then encodes the RNA bases pair sequence U, C, A, G, C, A in transcription. During the cell cycle, DNA molecule must be replicated. So at the end of the cell cycle, when there's 2 new cells, each cell receives a set of DNA. The genome is the complete set of DNA within an organism. And genes are DNA sequences that encode specific proteins that are transcribed into RNA. Not all genes are expressed in all cells of an organism, however. For instance, the genes that are activated in your digestive cells are not going to be necessarily active in your nerve cells. Our bodies have ways to regulate these genes. This is a nice picture to summarize what we went over in terms of DNA producing RNA. As we talked about, DNA can replicate to form new DNA, and then it can participate in transcription. During transcription, the DNA will produce RNA, which will then go into producing proteins. So as we mentioned before, this picture summarizes the roles and function of DNA. In Chapter 1, we talked briefly about evolution. With the study of DNA over the past 60 years or so, many evolutionary relationships have been revealed using DNA sequencing. Closely related species has been found out have more similar base sequences than species that are distantly related. Scientists often use genomes to compare organisms with one another to determine evolutionary relationships. Table 3.1 here is a nice review of some of the differences between DNA and RNA. I recommend to review this for your notes. Note the difference in the sugar and bases between DNA and RNA. Alright, here's our first progress check. Feel free to look back at the previous slides to try to answer these questions. Once you are ready, move on to the rest of this lecture. Next half of this lecture, we're going to focus on proteins. As we stated earlier, DNA encodes for proteins. So that is often why the two are paired together. Proteins have some major functions within the body. They are enzymes and participate in reactions producing products that our body can use. Proteins also play a role with antibodies. Proteins also help regulate physiological processes such as hormones and other regulatory proteins. And proteins can also be found on the surface of cells as receptor proteins and can receive and respond to molecular signals. There are also storage proteins that store amino acids. Some other functions of proteins are that proteins may play a structural role in the physical stability of structures. There are also transport proteins that carry substances such as glucose across the cell, or hemoglobin carrying gases within our blood. And finally, genetic regulatory proteins that help regulate gene expression. Over the course of this semester, we will focus on enzymes and touch upon some of these other jobs that proteins do. Amino acids are the subunits that make up proteins. All amino acids have a carboxyl functional group and an amino group that allow amino acids to have both acidic and basic properties. There's also what is called the R side chain that all amino acids have as well. All these groups are connected to a central carbon. All amino acids have the carboxyl and amino groups. The thing that differentiates the 20 amino acids is the R group. Amino acids are grouped according to this R group, such as being nonpolar or polar amino acids. This R group gives the amino acid its unique properties. The next 3 slides will go over the 20 amino acids. You're not required to know the amino acids. However, I do want you to know that each amino acid has an amino group and a carboxyl group with an R group. That R group is what differentiates each amino acid from one another and gives its unique properties. Notice, again, that all amino acids have a carboxyl and amino group, and they differentiate with their R group. Here are the last 7 amino acids. Do any of them look familiar to you? Amino acids make up proteins, as we've already mentioned. A peptide is a short polymer with only 20 or fewer amino acids. Some hormones and signaling molecules are classified as peptides due to their small amount of amino acids. Polypeptides are large polymers. Functional proteins can be made up with 1 or more polypeptides. Here are some examples in variation in protein length. Insulin only has 51 amino acids, as illustrated on the top left, while a muscle protein, titin, has over 34,000 amino acids. Amino acids bind together in condensation reactions. An OH from the carboxyl group and a H from the amino group bind together, forming water. And this binds the 2 amino acids together. This is called a peptide bond. This slide is illustrating 3 amino acids binding together in 2 condensation reactions. Water is a byproduct with each peptide bond formation. Next, we are going to talk about protein structure. Depending on the size of the protein, it may have a relatively simple or complex structure. They can have up to 4 different levels of structure. Not all proteins will have all 4. Again, it depends on the size. The first structure that all proteins have is what is called the primary structure. The primary structure is the sequence of amino acids that make up the proteins. You can see this on the top left of the slide. Again, primary structure is just the sequence of amino acids that make up the protein. Again, here's just another illustration of the primary structure of a protein. It's just a sequence of amino acids. The secondary structure of a protein is when there are regular repeated patterns forming regions. These regions are being formed via hydrogen bonding. Amino acids may hydrogen bond with other amino acids and may form two different types of structures, such as what is called an alpha helix or beta plated sheet. Here's an example of a secondary structure of a protein. If you look closely, you can see the individual amino acids from the primary structure. The amino acids are forming hydrogen bonds with one another, creating an alpha helix on the left and a beta pleated sheet on the right. After the secondary structure is formed, polypeptide chain can be bent and folded, which results in a definitive 3D shape and forms what is called the tertiary structure. The primary and secondary structure help form the tertiary structure. Here on the left, we have an illustration of a tertiary structure. You can see the beta plated sheets and the alpha helix. Here's the alpha helix and the beta plated sheets. They are bent together, forming the tertiary structure. Interactions between R groups of those amino acids determine the tertiary structure. R groups may form what are called disulfide bridges, or hydrogen bonds, or hydrophobic side chains. Here's a 3D model of protein tertiary structure. You can see alpha helix, little corkscrew looking structures here, and then the beta plated sheets represented by the red here. This is all possible due to the interaction of those amino acids R groups. Okay, here's where things can get a bit crazy and a little bit complicated. Sometimes proteins have a fourth structure known as the quaternary structure. The key word here is, for talking about the quaternary structure, is what are called subunits. When two or more subunits come together, they form a quaternary structure. Sometimes students will confuse the quaternary structure for tertiary structure. Remember, tertiary structure is made possible due to the interaction of R groups. For a tertiary structure, we're looking here at subunits, which we'll describe and talk about here briefly. Each of the units in a tertiary structure is made of alpha helix and beta plated sheets. These subunits are bound together by hydrogen bonds and hydrophobic and ionic interactions. Hemoglobin is an example of a protein with subunits. You can see hemoglobin at the top of this slide. Notice the top dark brown color and the lighter color on the bottom. These are two subunits of hemoglobin. So this top brown color would be one subunit, or this bottom lighter brown color would be another subunit. Not all proteins have a quaternary structure. On the right here is another illustration of a protein with a quaternary structure. Again, notice the areas that are labeled subunits. Those subunits are made of alpha helixes and beta plated sheets. If you look at the left here, you can see a protein that's in its tertiary structure. These tertiary structure proteins often become the subunits that make quaternary structure of other proteins. So why are we learning all of this? We learn about the importance of many enzymes, especially when we learn about the cell. Proteins have very specific shapes for their very specific role. These shapes and the role they play are determined by the amino acids R group and what they bind with. This can change the tertiary structure and shape of a protein. Proteins can also covalently bond to their own amino acids, further changing their shapes. Basically, the shape of a protein all comes down to the amino acids and how they act with one another. In the next lecture, we'll see how proteins can change their shapes. The shape of a protein will determine if it can participate in a reaction or not. The shape all starts with the primary structure of amino acids. Proteins can become denatured. Denatured is when heat or a chemical disrupt weaker interactions in a protein, such as their hydrogen bonds. Denaturing can destroy secondary and tertiary structures of protein, thus altering their shapes. The protein may return to normal when cooled or the chemical is removed. All the information needed for the shape of the protein to come back together is contained in the primary structure. Some factors that can cause denaturing are temperature, which usually happens with an increase in heat, or a change in the hydrogen ion concentration. This can alter the pH. Polar substances can disrupt hydrogen bonding, and nonpolar substances may disrupt hydrophobic groups. All this can cause denaturing of a protein. With that, we finish up part 1 of Chapter 3. Please take your time to answer these questions. We covered a lot, so be sure to take your time with the material. Thank you for listening, and have a good rest of your day.